For so long, we've thought about the cerebellum. It really started as kind of a fantastic anatomical structure. But then, as kind of modern day science shifted, we started to ask, well, what aspect of movement is it involved in? Is it involved in initiation, planning, the execution of movement? So that was kind of the debate for a long time. And then in the last 15, 20 years, the cerebellum has been thought of as a structure involved in cognition as well, whether it's emotion, whether it's decision making.
Dr. Roy, welcome to the podcast.
Thank you very much. It's wonderful to be here.
I'm so excited to dive into what we were just talking about before we started this podcast was how underrated the cerebellum is. And you said that you've been saying that for 30 years now. So tell me a little bit more about that if you don't mind.
Yeah, not a problem. So, for so long we've thought about the cerebellum. It really started as kind of a fantastic anatomical structure. And that was what it was a great model for. And, this is Ramon y Cajal's work, which his Nobel Prize, a lot of the work which he loved to cite was based on cerebellar anatomy and what he found. And then what also his competitor and Nobel Prize winner, Camillo Golgi, also found was they both studied cerebellum. So decades went by, 100 plus years, and then it became very clear from a lot of the physiology and clinical science that the cerebellum may be involved in movement. But then, as kind of modern day science shifted, we started to ask, well, what aspect of movement is it involved in? Is it involved in initiation, planning, the execution of movement? So that was kind of the debate for a long time. And still, we don't actually, shockingly, we don't have all the answers to that. And so with, of course, the importance of the cerebral cortex and other subnuclei, basal ganglia, are also involved in movement. This is when I say questions re-arise and people say, well, what's the cerebellum actually doing? And is it important for this? So, and then the question really goes down to, well, what else is it doing? And then in the last 15, 20 years, the cerebellum has been thought of as a structure involved in cognition as well, whether it's emotion, whether it's decision making. And now that question even gets bigger. And people start to think, well, scratch your heads now after hundreds of years of studying this structure. And you think, well, what is it doing? And how is it doing all of this? And is it even doing any of it? People start to ask, because there's cases where people are actually born without a cerebellum, shockingly. And even though these people live, they have a lot of movement problems. They have a lot of problems with decision making. Intellectual capacity is compromised. But it's actually surprising to people. And I think our view is that when we say something is important, what we really want as human beings, as scientists, is that if it's not there, you shouldn't be able to do anything. And in fact, this is where you start to think about connectivity within the brain. And I now tell people, well, okay, hold on a minute. It's very important for movement. We think it's very important for cognition, but it doesn't operate alone. And if it's damaged or if you develop without it, there's other regions of the brain that can compensate for some, but not all of its functions.
Fascinating. And what really caught my attention was the amount of neurons and the amount of synaptic connections across
Yes.
each neuron in the cerebellum.
Yeah.
So like this first principles thinking says, well, yes, it has to be important. Why
Right.
otherwise, why would it be so dense? Why would it have so many connections? So that's where my mind went.
Yes. Yeah, absolutely. And this is something it's hard to grasp even when you look under the microscope, how many cells are packed in there. And it's mainly due to one cell type, the granule cells. This is the one overwhelmingly large numbered cell type in the cerebellum. And this is the single cell that constitutes more than half the entire neurons in the entire brain. It's just incredible. And in fact, because of its numbers and exactly what you're saying, about 20 years ago, there were several hypotheses that suggested, well, we have a lot of granule cells, and it's really built into the brain almost as a reserve, almost that you can never run out of granule cell function because you have so many. And then people theorize that, well, we think you probably only use about 10% of your granule cells. Well, and this also was in the rest of neuroscience. People thought, well, you're only using 10% of your brain. I've heard that many years. Part of that is, and I'm glad you're shaking your head because I don't think that's true. I think the only kernel of truth one might think about in a statement like that is that in any moment in time, and I'm talking any millisecond, and you may only be using a portion of your brain. And that makes sense to me because you're not always doing the same function for every single thing throughout your entire life. But it's so dynamic that in a single second, you're probably using your entire granule cell population. Right. So this is what I think it's just mind-boggling how many. But I want to add something to disease because for me, this is what hits home when you think about the number of granule cells and you toggle with what is already so voluminous is medulloblastoma. This is the most common pediatric brain cancer. And it takes a tiny bit of misregulation of granule cell proliferation during development and you spark a medulloblastoma, a very aggressive cancer. You already have so many granule cells, and they're proliferating like crazy. Starting embryogenesis, the progenitors start to divide and migrate, and then postnatally, you have this second wave of neurogenesis. And that is what produces the large number of granule cells we know. But there's several genes that are required for normal development and normal proliferation. And if they are misregulated, we know this happens in cancer, now you have so many more granule cells, and of course, you get a tumor. These numbers are shockingly large, and unfortunately, if you misregulate them, you end up with a very, very severe childhood condition.
Interesting. And can you break down the difference between like a granule cell and a Purkinje cell in the cerebellum?
Yeah, so one of the big difference I will start with is that granule cells are all excitatory neurons,
Mhm.
and they use glutamate for their neurotransmission, as opposed to the Purkinje cells, which are GABAergic and they are all inhibitory.
Mm.
The two of them, once they connect, have this beautiful balance of excitation and inhibition that we know is operational throughout the nervous system. You need that beautiful balance. The other big difference between them is that the granule cell, and this is actually kind of special for the cerebellum, the way it's organized developmentally, the
Hmm.
granule cells are born in a very different place to the Purkinje cells. And it's pretty cool, actually. So the granule cells are born in the posterior aspect of the cerebellum. It's a little region called the rhombic lip. And they will proliferate in a first wave. They'll migrate over the surface of the cerebellum. The Purkinje cells are born underneath in the ventricular zone, and they
Hmm.
will migrate upwards. And then once the granule cells hit a second wave of proliferation, then they start to differentiate and migrate down, and they kind of bypass each other. And as they pass each other, the granule cells will send an axon and connect to the Purkinje cells.
Wow.
And now you start to form the essential microcircuit of the cerebellum. The other big difference is that the granule cells, because of this large number, again, this is mainly a theory right now, a big difference, they're kind of thought of as a filter of information because they're going to receive tons of sensory information from the spinal cord, from the brainstem, from the cortex. It's all coming in and into granule cells. The Purkinje cells are the primary computational unit. They're going to then receive all the filtered information, compute and make sense of those commands, and then pass it on downstream. So they're born in different places, they use different neurotransmitters, and they actually have very different jobs, but they function together, very tightly knit. So much so that if in diseases, whether you have different types of injuries, ataxia, if you kill Purkinje cells, you end up killing granule cells. And we
Mm.
also know that developmentally, if you have a disorder that kills granule cells, you end up killing Purkinje cells as well.
I see. And another reason that's created my fascination with the cerebellum was people that do have ataxia or
Yeah.
they have had damage to the cerebellum. Now,
Yes.
when it comes to helping repair and get the cerebellum working better,
Yeah.
what at this your research center? What do you find with the neuroplasticity of the cerebellum?
I've been through so many different phases in my career trying to attack how do we fix the brain? How do we fix disorders? How do we repair movement? And particularly, again, I've focused mainly on movement over the years.
Mhm.
And I've been through trying to understand molecular tools, so molecular therapies. We've been through trying to understand, and actually, I started as an undergraduate, I was actually a kinesiologist. So I actually worked in physiotherapy clinics looking at mainly rehabilitation due to workplace injuries. And at the time, I was more interested or more involved in understanding muscles, but little did I know at the time that type of rehabilitation is actually driven by the brain. You are
Mhm.
making the muscles work, but it is all the brain. And you said the key word here is neuroplasticity. And the reason this is so fascinating is that over the last 10 years, been focusing very heavily in my lab on deep brain stimulation. I've been trying to tackle this problem of how to repair circuits and how to repair when things have gone wrong during development and adulthood. And deep brain stimulation has been such a fascinating topic for me. And our most recent work, and much of which is unpublished, but we just had a nice review that will come out very soon. We think the benefits of deep brain stimulation are rooted in plasticity.
Mhm.
And the reason we were very excited to find this is that the deep brain stimulation approaches that we've developed in my lab actually put the DBS electrodes into the cerebellum.
Mm.
Previous DBS puts the electrodes in the basal ganglia, different areas, even the brainstem, in the thalamus. We chose the cerebellum not only, as we've been talking, because of this massive capacity of the amount of circuits and cells and connections, but I already kind of, if you will, I was kind of cheating because I already knew about plasticity in the cerebellum. And what we're seeing now is that the type of changes that we might be inducing to repair circuits in ataxia, dystonia, and tremor may be plasticity driven.
Mm.
And these may be the very same or very similar mechanisms to what happens when you use physical rehabilitation. For many years, since I was an undergraduate, literally going back to my teenage years, the idea of rehabilitation and combining that with whatever therapy you might have, rehab and physical rehabilitation, and now even the mental types of rehabilitation, I think these have to be part of every regime for repair because that taps into neuroplasticity.
That's amazing. I've heard so many good things about deep brain stimulation, and in fact,
Yeah.
a colleague and friend of mine worked for a company, and that's basically the essence of what they did.
Okay.
And
Yep.
he was it was transformative for him too, because he witnessed people that were having extreme tremors from
Yeah.
Parkinson's,
Yes.
and then they do it, and it's the tremors stop
Stops. Yes.
like a miracle pretty
Yeah.
much. And that's unbelievable with the cerebellum and the opportunity that is there. So the next kind of deviation I'd like to go to with the cerebellum, I we're kind of guessing here because we the brain is so mysterious,
Yeah.
but we think like the cognition and memories and just kind of the outside the box things that they're
Yeah.
discovering about it. Uh, can you dive into maybe memory right now
Yes,
in the cerebellum?
absolutely.
Yeah.
So, this is one of the things we've always pinned the type of information in terms of memory that the cerebellum is operating in is really motor memory. So, motor skill acquisition. If you get good at something, let's take tennis, for example, because it's a nice repetitive in terms of the training. You have to be very precise. Error correction has to be very good. Reaction time has to be fantastic. And you get good at that as a tennis player. But let's say you have some kind of injury or you stop playing for a while and things slightly degrade, right? Once you start training again, you are much quicker to get back to the level you were than you were as a novice because there's memory in the cerebellum. And in fact, with all of these granule cells, the prediction now with electrophysiology data suggests that almost the way memory is encoded in these granule cells, it almost has these kind of lookup tables that it stores. And for all of these different versions of movements and dynamics and initiation and planning, there's these lookup tables in different channels, and the cerebellum can pull those down and get you back to where you were much quicker than you were. And then we always thought of the other part of memory, remembering people, remembering things that we value in our lives. That was really stored in the hippocampus. But that's changing. There's several labs now that are showing through several connections, and we don't know the anatomical pathway, that the cerebellum actually communicates very heavily with the hippocampus. So now we have to redefine, well, what are we talking about when we think about a memory, and where is this stored? Because let's take the event of, like I said, let's say, you took some beautiful trip to Italy with your family. You're going to remember that. And we think that's encoded in the hippocampus because it's a lot of emotional regulation. There's something very valuable that you store. But some of that information is embedded in motor sequences. So you must have the cerebellum to actually help the hippocampus back and forth, encode that information. And a slight deviation of this, a colleague of mine, Detlef Heck, he's now the chair in Minnesota, in Duluth. We had this really cool study that I have to give all the credit to Detlef for thinking about it. He's a cerebellar guy too, but he also thinks about the cortex and other regions. And in some of his experiments, he realized that in mice undergoing a paradigm for decision-making, it was more than just the hippocampus, and there was already thoughts that the prefrontal cortex was involved, and the two of them, with the activity, there had to be some kind of coherence and regulation between the two. But in some of his initial studies, he realized that if he manipulates the cerebellum, the coherence activity between the hippocampus and prefrontal cortex was not as it should be, and decision-making was abnormal. When the animal was asked to make the right decisions over and over, it started to make less correct decisions. And so Detlef called me, he's like, hold on, wait a minute. This kind of encoding of memories and decisions and moving forward, if you will, there seems to be multiple very big nodes that we've always all thought of as memory for this aspect and memory for that aspect. And now the cerebellum is defining and driving this non-motor type of information, which again is rooted in this whole idea of memory. Well, it turned out if we use a tool called optogenetics, where we genetically encode these channels which are sensitive to blue light, and we can control activation of neurons. If we put them specifically in the cerebellum, and we shine the light at specific times while the mouse is trying to make decisions, we actually alter the coherence in the circuit, and the mice make poor decisions. So that was just a really, really beautiful example of how, as your question about memory, we now have to rethink not only how we think about memory, but where memory is encoded and controlled as well.
Once again, so interesting. And I think about this a lot, is that the brain is all about energy conservation.
Right.
And when it comes to learning. And so my fascination is I'm just throwing this out there, but to me it makes sense, is that initially, like, we have conscious experience in
Yeah.
our brain's prefrontal cortex and frontal lobe, and then essentially the information perhaps could get sent to the hippocampus and the
Yeah.
limbic system,
Yeah.
and eventually, maybe with enough effort, it gets stored away in the cerebellum,
Yes.
like one, two, three,
Yes.
as a way of conserving energy. I don't know what you think about that.
I love that idea. You know why I love that idea is because you remind me of something the cerebellum is very good at doing. It's very good at giving us the ability to do things on automatic. And I'll even take it a step further with my analogy, because I can drive stick and I can drive automatic. And driving automatic is way less energy demanding. So if you think about your cerebellum as, kind of this automatic thing, and
Right.
I love what you're saying. If eventually the information is there, and the cerebellum can just run with it and make tiny adjustments that may or may not have a tiny influence from the prefrontal cortex, limbic system, just trying to help the cerebellum do this, do that, don't do this, and then the cerebellum does it and somebody else corrects it. I love that idea, actually.
Yeah, that's cool. And so I think of pathways perhaps, and I think the opposite is true as well, that it's sending a signal back and forth like,
Yeah.
and there's all this connection going on, and uh yeah, that conservation of energy. Recently, I learned about walking, is that why walking backwards is so powerful. It's because
Right,
think of the amount of repetition that we have in our lives walking forwards, and the
right.
cerebellum automates that process
Yes,
to make it
yes.
easier. So, yeah, maybe walking backwards is something people can do more of.
Yeah, absolutely. Yes. And you know what's fascinating? You remind me of something, and it may or may not be related, but there's some thoughts, and some people may or may not believe this, but in dystonia, which is a problem of muscle contractions, there's two things, at least two things that'll define somebody with having dystonia, is that you have abnormal co-contractions of agonist, antagonist, and antagonistic muscles, or you have overcontractions of your muscles. Either way, movements are disrupted, and certainly locomotion is problematic. But there have been now cases where people walk backwards and the dystonia disappears temporarily.
Wow.
And I can't explain it right now, but and initially when people have seen this, and you can see some of the stuff on YouTube, and some people say, oh no, they're not true. But actually, if you talk to some neurologists, they have actually seen that.
Hmm.
And I'll even put something even more dramatic, which many years ago, and this goes into, we know that mice are not humans, and we know that animal models, we have to study them with very particular focus on what we actually trying to study. We never, as scientists, want to make a mouse a human. It's about trying to understand mechanisms and fundamental principles in science.
Hmm.
However, we have created mice with dystonia, which have taught us a lot over the last 15 years. One of the things which was anecdotal in my lab, and it's something I always just kind of brushed off, very often my students and postdocs would come to me and say, when I'm watching our mice with dystonia, many cases they start to walk backwards.
Huh.
And it may be that there's an energy demand, like what you're saying. They're basically trying to find the most energetically favorable sequence of motion. They go forward, they have dystonia, they go backwards, it might be relieved. And again, I don't know where it sits in what we're initially talking about, but this idea of actually moving backwards, locomotion, and having a different sequence of energy, there may very well be something in this.
Yeah, and my mind goes to athletes, in how they can utilize the cerebellum, for even more improvement, better reaction
Yeah,
timing, and
yeah,
things. Have you all
yes.
done any research around like extreme high levels of performance in the cerebellum?
Well, yeah, you remind me of one particular study we did, actually. It's related to tremor. So we've been trying to figure out, it started out as a simple question because for the last 50 plus years, people have really thought the cerebellum is a key site for tremor. Again, it's not the only one, but we think it's a key site. But if it is a region involved in tremor, well, what are the cell types? So one of my students at the time, Amanda Brown, she's now a postdoctoral fellow in the lab, she went through a series of genetics and optogenetics and pharmacological studies, and we defined that the Purkinje cells can drive tremor through its connectivity with a brainstem region called the inferior olive. Okay,
Hmm.
and that was great, but that was also something that was heavily predicted based on physiology. Fantastic. But then came my next student, Joy Zhao, who then asked the question, well, once we get tremor, why is it that the drug propranolol, which has been used for several decades, can be very effective in tremor? Is it actually affecting the cerebellum? So she then went and did a series of genetic studies and electrophysiology studies and showed that yes, propranolol does impact the cerebellum and it does reduce tremor by remodulating activity in the cerebellum. But why am I telling you this story? When you asked about athletes, well, it turns out that when we do these studies in normal mice, and we all have a certain baseline tremor, tremor is very important for activation of muscles. and we all have a baseline tremor. And it turned out that propranolol actually lowers the amount of tremor
Hmm.
in normal mice. We were shocked to find out, and Joy discovered that there was one or two papers that Olympic athletes in certain countries were getting banned from using propranolol. And there's a particular paper that shows it was the shooting. Well, it turns out that if athletes take propranolol, it reduces your tremor, it may improve your reaction time, and it certainly improves your steadiness using a gun. So it became a banned substance in certain countries. Isn't that incredible?
That's crazy. That's crazy, man. Yeah, that's wild. I can't even believe that. And
Yeah,
it
I
does
was
make
really shocked to find that.
and it makes you wonder, like, I wonder if they test for that in like if that's something that they test for in like soccer or
I don't know,
things like that
actually. You know,
or
I mean,
baseball.
anything that requires precision, if you're able to lower that even a tiny, tiny bit, really is beneficial.
What other stuff were you involved in your research center, and how long have you been doing that, by the way?
Oh, this is, I'm so happy you asked me that because this is something that means just the world to me. I've always been in the cerebellum, and once I started as a graduate student, I never left the field. And despite, some mentors saying, you should do your postdoc in this different field, you should learn more, and I totally see the value of it. But I was just like, I'm studying cerebellum. I'm never switching. Um,
Good. Yeah.
And so some of the, so for a very long time, I had this dream of having a cerebellar center because as I learned more and more, and as we've spoken about today, so many things can go wrong with the cerebellum. It has so many functions. It contributes to so many disorders and diseases, whether they're genetics, whether they're injury, whether it's a concussion, whether it's alcoholism, or maybe it's aging. The cerebellum is always right there with other brain structures of importance.
Hmm.
So a couple of years ago, I was talking to my director and saying, okay, I'm super excited where I've gotten in my career. I'm very grateful for all the students that have come through my lab, all the opportunities, but I wanted to do something big for kind of my second half of my career. And as we were sitting, her and I were sitting at lunch, and she said, have you ever thought about putting together a group of cerebellar people here? And I said, actually, I have. And not just a group. I said, we need a center. And she said, wow, okay, that's big. And I said, I want a multidisciplinary center. It's not only basic science. I don't want to just solve the cerebellum, but I want people involved in neuro-oncology, rehabilitation, our neurosurgeons, our neurologists, our neuropsychiatrists. And I said, we're in Houston, where we have this medical center is literally the biggest medical center in the world. And I said, I know that it's not just me and you that are involved in cerebellum. And I'm talking about my director. Her name's Dr. Huda Zogby. She's actually the one that discovered the gene for spinocerebellar ataxia type 1, SCAR1. She was also the one that discovered the gene for Rett syndrome, the MECP2 gene, which also has a cerebellar component. So Huda's been like a cerebellar person since her early days. So it's like, Huda, it's not just you and I that are interested in cerebellum. There's so many people in Houston. So what we were able to do is put together this Cerebellar Science Center across 9 different institutes and hospitals across Houston. So we have Rice University, we have Baylor, Texas Children's, Houston Methodist, Texas A&M, UT Health. It's just incredible. I have faculty at University of Houston just up the road. It's been just a remarkable a start to a wonderful collaboration. And I've tried very hard to dissolve the boundaries of academic institutes. And I said, well, this isn't about money. It's not about papers. This is about solving tough problems that will solve science questions that have been boggling us for hundreds of years and solve disorders and conditions and problems that come up time and time again. And usually it's 1, 2 labs tackling this. I said, I don't want just a couple of labs. I want, if somebody comes to us and say, there's this problem with a child and there's this new gene and this mutation, can you do something? I want to be able to assemble a team of 10, 15, 20 labs and say, we got to solve this as quickly as possible. That's kind of where I'm headed, where my goal is. So we have this center, we're off to a great start. And I'm trying my best to step outside of my comfort zone and not just, one of the things with doing something like this, all the scientists like yourself, we all know science is important. We all know neuroscience is important. We all know cerebellum is important, but the rest of the world may not know that.
That's right.
And so I think this is a really good opportunity to teach people, educate, and collaborate with the community, and to actually also understand, because as myself as a scientist, I'm not only driven by what's cool and exciting in biology.
Yeah.
I am super motivated by what is going to help somebody. I really am, truthfully, that does motivate me in the lab. And almost all of our questions now directly come from patient problems, even though we're a research lab. So we are guided by real world problems. And this center was to motivate my colleagues and come with me on this journey.
I love that. I really do. And I've got just a few more questions before we end this conversation. The one right now is if somebody that is dealing with a cerebellum disorder, whether it's rare or
Yeah.
whether it's from damage,
Yes.
or what would be like some encouraging words that you could give that person?
Yeah, so the encouraging words I would say is that we have finally hit the point in medicine, in science, and in neuroscience specifically, that if you have something, there's always a starting place. We now have things will get better, but we have a way. Let's say like you said, you have a mutation. We now have, even here at Baylor and Texas Children's, we have something called the Undiagnosed Disease Network, which is a nationwide effort funded through the NIH, where if you have a mutation and you don't know what it is, and your child or you is struggling from this, we can now use a combination of Drosophila genetics, in vitro cell biology, in vivo, and clinical perspective with clinicians, not only to pinpoint the mutation, but identify the gene. Once they identify the gene, now we have in place that you can actually, if it's a neurological problem, we can now talk to neurologists, and we have so many competent neurologists around the USA and our colleagues around the world, but we have so many good doctors in our country. We are incredibly lucky and privileged that we are now in the case where you can talk to people not only at one institute, but multiple. Then you can start to consider, well, what type of rehabilitation is there, if it's available, and if it does any good potentially for your condition. Then you can start to think about, because there's many trials on now, whether it's different molecular therapies, whether it's different drug therapies, there are actually many trials going on. And I do know what we see in the news is only this failed and that failed and this failed. And that is true, but I think it's also unfair, not only to the public, but also to science, because we have a lot of big wins every day. But I think we need to talk to each other more. So when people arise and they ask questions, they're going to find out that people are working very hard to try to solve these conditions. And after that, again, there's also different foundations. I don't want to forget about the value of different disease foundations, whether they're small foundations or big foundations. The patient support that they have, the type of information that's being collected is very valuable. Genetic information, I know several foundations are working with the NIH to catalog and release this information so that the geneticists can know and track these different problems. And also there's population genetics. We need to see where these different mutations are arising. Where are they and what are people doing? So again, the different disease mutations, the different disease foundations have been a very critical collaboration for the scientists and clinicians. So if somebody comes and they have something going back, there's a lot of hope and there's a lot people can do. And from rehab to molecular therapy, and as we talked about, if you might be a candidate for either deep brain stimulation or maybe non-invasive stimulation, like transcranial stimulation, or there's other forms as well. There's a lot of consideration and a lot of conversations to have with your doctors.
How have you integrated like cerebellum work in your own life, like on a day-to-day basis?
Yeah, so from a day-to-day basis, I have to say I've always been into weightlifting because I wasn't very good at soccer. I was terrible at basketball. and many other sports. We did okay as kids as swimming, but then I discovered that I really loved weightlifting. And for my own day-to-day life, and the reason I mention this is that I am actually a candidate who was born with some kind of developmental mutation that I have severe back problems. Based on MRI and some really careful X-rays, it turns out that I was born with an anomaly where my vertebrae are fused to the inner walls of my pelvis.
Mm.
And they're fused at an angle. So I've always had these extruded herniations of my discs, which is very painful. And the reason I'm saying that, my love for exercise and herniated discs create a very problematic lifestyle, right? So my understanding of the cerebellum and movement and timing and plasticity, trying to understand how does your body respond appropriately, how much movement is good, how much movement may be damaging, and are you able to use the cerebellum and the principles of neuroscience and plasticity to actually improve my strength, my agility, and also muscle mass as well, because it's not just being jacked, I should say, but actually preserving a certain amount of muscle mass as you age is very important for
Hmm.
your whole body, for your bone structure. So for me, it's all based on what type of information is the cerebellum processing. And when I design my exercises, it's not just go in and lift heavy. It's actually, I'm trying to have a lot of mindfulness of sensory information too. I think proprioception is very important, clearly to the cerebellum, but the way you actually design an exercise routine, your warm-ups, you have to be, you have to think about joint position and full range of motion. All that is basic biomechanics and how it interacts with the cerebellum. And I use that every day in my life.
Awesome. That's super cool answer. And number one, thank you for coming on. And uh, how can people reach out to you? One, and what is just the last thing that you'd like to say to anybody that might be listening?
What I would love to say is that without a doubt, the most exciting time of science that we have been in history. And I don't care what aspect of science, I am a neuroscientist and I just absolutely love and I'm passionate about the brain. But there's many other areas of science where we've all done just incredible efforts to solve big problems. And that has translated into hope for better therapies, for better treatments. And I say this very carefully because it's not a word I like to use too often, but I think we can start to think about little glimmers of hope for cures of certain disease. And that's always our driving force. But again, I use that word very carefully because finding a cure for any disease, this is not a trivial thing. And I don't like to just throw that word out there. But I think where we've come in science, we should all have a lot of hope for where we're going. And if people want to get a hold of me, I think the best way is to go to our Texas Children's Duncan Neurological Research Institute website. I have my faculty profile on there, and you'll see everything we're doing in our building and what I'm doing in my lab. And very, very soon, we will have our own website embedded in there for the Cerebellum Science Center, so you can see what we're doing there. But my home is at Texas Children's and Baylor, and you can find me there.
Congratulations on all of that, and thanks again for coming on.
Thank you very much. This is a great pleasure talking to you.